Today, we will be discussing oxytocin receptor allelic variation, neuroplasticity, and online learning environments.
My name is Nishmaram and I'm from Florida.
Hi everyone, I'm Avan Sapkota and I'm from Nepal.
Hello, I'm Coral Grice and I'm from Kentucky.
Hello, I'm Lillian Politz and I'm from Florida.
Hello, I'm Redeema Thacker and I'm from New Jersey.
Hello, I'm Sahana Rajagopalan and I'm from Delaware.
Hello, I'm Som Kisti and I'm from France.
We will now be diving into the background portion of the research proposal. Our impact of question. The COVID-19 pandemic has had a huge impact on how students receive their education and, more importantly, how they perceive this information. However, the impacts from student to student may not be the same. Let's look at some examples. Let's look at Lisa. She was an undergraduate student in college when March 13th, 2020 hit. She was a very social butterfly. She loved hanging out with her friends. She was captain of the crew team. And when COVID hit, she found it very hard to cope with the fact that she couldn't see her friends today. She is known as an extrovert. Now, let's look at Jennifer. She prefers to spend her time alone in her room reading a book rather than going to a party. She is what we know as an introvert. COVID has a different impact on different students' personalities. This gives us an opportunity to study the brain even more at a new angle that we wouldn't have had the opportunity to do before. Out of 5 million students surveyed in the US Census Bureau, 42% of these undergraduate students experienced major difficulties with concentration and motivation, whereas 37% found it to be minor. We are looking today at what influences people to adapt to different learning environments, which in this case is the oxytocin receptor gene. We are also looking at the susceptibility to neuroplasticity and wanting to look at the allelic variations with the increased ability to adapt. We're discussing susceptibility to neuroplasticity since there are certain genotypes that make people more sensitive to certain situations.
Now, because of what we know about how oxytocin influences social behavior and social cognition, it can be said that the molecular variability in the gene that codes for the oxytocin receptor, or OXTR, would affect human complex social behavior. Specifically, the single nucleotide polymorphism of RS53576 is a silent G to A change in the oxytocin receptor gene. Those with the major G allele have an elevated sensitivity to environmental factors, and with that comes a greater susceptibility to neuroplasticity. It's also important to note that the major G allele promotes the efficiency of the oxytocin system, which also plays a role in causing this greater susceptibility.
Oxytocin receptors are associated with neuroplasticity. Prior research asserts that neurons constantly undergo structural and functional alterations as a part of a process called synaptic plasticity. Therefore, there is no doubt that oxytocin could affect synaptic activity at many levels of regulation. For example, oxytocin-containing neurons have an impact on neurogenesis in several different brain regions. Specifically, oxytocin plays a role in expression of neurotrophic factors, BDNF, and in nerve growth factor, both of which are involved in neuroplasticity. The gene variance in oxytocin is a critical factor in organizing neural circuitry and social behavior development. This brings about the question, what is neuroplasticity? Functional neuroplasticity involves physiological changes in nerve cell function, such as the frequency of impulses or the probability of release of a chemical signal, both of which act to make a nerve impulse weaker or stronger. And it also creates changes in the degree of synchronicity among a population of cells. Structural refers to volumetric changes in discrete brain regions and the formation of new nerve fibers or branches, or by the growth and addition of new cells. Different genotypes have different vulnerability to plasticity. For example, the AG versus GG alleles have different vulnerabilities to the amount of plasticity in a person's brain. So specifically for OXTR, people have a selective advantage for certain environments if they have the GG allele. Therefore, environmental contingencies may be beneficial or non-beneficial for certain individuals depending on their genotype. Here is a complex example of how oxytocin plays a role in the brain and in specific cells.
So what is unknown? We know that oxytocin influences complex social behavior. However, we don't know whether that behavior can be negative or positive all the time. A study done by Jennifer Bartz et al. Found that oxytocin can actually hinder trust and cooperation in borderline personality disorder and actually make those symptoms come to life. So that is something that is still unknown.
The question that we will be exploring is, what does oxytocin receptor allelic variation predict about adapting to an online learning environment?
Now, in terms of our aims and hypothesis, our first goal is to understand if the genotypic variation seen with OXTR could affect how much our test subjects' academic performance changes after the shift from in-person to virtual learning. We hypothesize that students with the A allele, whether it be AG or AA, which is associated with decreased sensitivity to environmental conditions, will have greater changes in academic performance, whether it be worse or better based on learning environment comfortability.
Aim 2 questions. Does introversion and extroversion account for differences in GPA, changes within the genotype groups? Presumably, the best predictions are, firstly, introverts will do better and the extroverts will do worse within a changed learning environment to online. This relates to the social development aspects of OXTR. Additionally, introverts traditionally thrive in less social scenarios. Connecting this to selective environmental conditions, in individuals with genotypes concurrent with greater neuroplasticity, socially stressful situations may vary. And secondly, OXTR type A introverts and extroverts will have greater increases and decreases in GPA respectively. Examining perceived isolation as a possible explanatory variable for variability within those genotype groups is very important. And the isolation rating will back up the toughness of online social isolation among groups of introverts versus extroverts. And these predictions will correlate with AIM-1 as well.
So now let's get into our proposed method of our research study. First of all, we'll start by selecting a middle-sized undergraduate institution. From that, we'll randomly select around 1,100 undergraduate students for our study. Exclusion criteria of sampled students include past history of diseases or substance abuse in the past 6 months. Next, we'll ask these students the permission to access their grade point average GPA of the 2 semester from the institutional registrar. GPA of 1 semester would be just before the onset of COVID-19 in a real class setting, and the GPA of a semester just after COVID-19 in an online setting. In our study, we set the criteria for GPA change to be 1.25%, and that would correlate to 1 arbitrary unit change in our study. After we have these GPA change of all the students, we'll divide students into 3 groups. Firstly, group 1. Group A, these students will have higher GPA in online learning compared to the in-person class. Secondly, we have group B. In this group, there are students who have lower GPA in online learning compared to the in-person class. And lastly, we have group C, students that had less than 1.25% change in GPA in online learning versus the in-person class. After this, we'll ask the students to fill the 2 forms. One is the perceived social isolation PSI form, which will help us assess how isolated they felt in the online learning. Similarly, we'll ask them to fill out the MBTI test, and we'll only look at the introversion and extroversion subsection in this test. We have also decided to incentivize them with an Amazon gift card. After that, lastly, in order to find out the allelic variation of the oxytocin receptor OXTR genotype, we'll get the student's saliva sample. Then we'll use a PCR polymerase chain reaction to find out which genotype do these students have, such as GZ allele or AZ or AA allele. After that, lastly, we'll analyze all this allelic variation, introversion and extroversion, and try to correlate it to the GPA change together. This is the procedural diagram of our study.
Moving on, now we will be looking at our potential and anticipated findings. So we expect to see students with the A allele who are introverted having a rise in their GPA, and introverted students with the G allele having a lower GPA. Also, we will have the expectation that students with the A allele who are extroverted will have a fall in their GPA, and students with the G allele who are extroverted will also have a lower GPA. But of course, we will expect to see some students with these alleles who are extroverted and introverted, both resulting in a higher or lower GPA.
Some considerations that we have to take into the study are the limitations. There are demand characteristics of participants' home life disturbing students' learning, and that is why students have decreased academic performance and not their genes. Epigenetics create a demand characteristic of varying gene level expression between subjects. Another demand characteristic is the possibility that the teacher's teaching style changed with online learning, and that is why participants' academic performance has changed. Money will be an incentive for participants to volunteer for the study, and because the sample size is large, the amount of money spent will be great. The probability of demand characteristics affecting the study is low because the sample size is so large that there'll be less of a chance that the majority of the sample size's home life disturbs their learning. Methylation is significantly lower in children and young adults, so there will be less of a varying gene level expression. Changes between online and in-person school are more significant than the more minor variances between types of online learning.
Now, let's talk about the ethical, legal, and social implications of our proposed study. The first implication I'd like to go over is the privacy of genetic sequencing. Without a doubt, the students' genetic data are needed for this study. However, this data could influence their futures if made public. It is possible that schools could discriminate against certain students if their genetic information was available. The second implication is whether or not schools should be held responsible to aid students depending on their oxytocin allelic variation. If we find a link between oxytocin allelic variation and academic performance in an online learning environment, is it the school's responsibility to accommodate students to an environment they might learn best in? The third implication I'd like to present is whether or not different environments disadvantage genetically varying students, and how will schools provide equity? Given the results we may find, how will schools manage to create an equitable learning environment for all their students, given their allelic variation and introversion or extroversion?
Okay, guys, so Morgan, you can now start.
All right, terrific. Thank you so much, you guys, and well done. Way to kick it off. That was absolutely amazing. You guys were so thorough. I appreciate how you really dug into the biopsychosocial. That was really great. So I'm so glad you are our first group. We could definitely use you as an example for all of the groups to follow. Well done. Mark, if we want to bring up the panelists now, and if someone wants to stop sharing their screen, then we can do feedback. All right, congratulations again, you all. You should feel really proud of yourselves. Well done.
An excellent job, a really excellent job. And what I'd like to do in terms of feedback is provide feedback from Dr. Rachel Wurtzman, Professor Dr. Nico Coles, and on style and presentation, Ms. Eleanor Thompson. Rachel, over to you.
Hello, guys. Absolutely great job with your presentation. You really fleshed it out. You were organized in your presentation order. The only I noticed that you might have gotten your signal crossed or somehow reversed your hypothesis a little bit with the type A versus the type G. But I think that that must have been a slight mix-up because everything else in your hypothesis and your anticipated results and your interpretation was internally consistent. Yeah, I just noticed that it was when we had talked, it was the allele that was associated with a greater susceptibility to plasticity that would have the greater changes. But your logic with that was dead on, and this was a really creative project. I'm very proud of you all.
Professor Dr. Kohls.
Yeah, thank you, Jim, and hello from Germany. First of all, I have to say I'm really impressed. I mean, this clearly shows that there is a lot of scientific rigor, will, and competence involved, and it is clearly visible to me that you have taken a deep dive into not only neuroscience but also methodology. I think you have well elaborated on the proposed details, sample size, and you've looked into questionnaire measurement instruments. You looked into the the foundations of neuroscience. So you are to be applauded. I can say. A question that came to my mind was, if I remember correctly, you calculated the sample size was 1,100 students, and I wonder why that is the case. Have you done an effect size calculation? And the second question is, obviously, you were referring to the Big Five model that was given to us by Costa and McCrae in 1982. And I saw that you were, interestingly enough, using another questionnaire, the Myers-Briggs Type Inventory, and I wonder why that was your preferred choice.
Regarding the sample size, we are just going through like our previous studies that have talked about genetic and environment interaction, like relation, and they presented that anyway between like 500 to 1500 was a good amount, and I think just like between 1100 would be like an ideal.
Mm-hmm. Okay.
And what about the Myers-Briggs? That is an interesting question.
Regarding the MBTI test,
yeah,
we just thought that if people are like introverted or extroverted, that also has a role on how like they take like some sort of social stressors. Like some introverted people might not have like felt that they were much more isolated in this online learning than people who are extroverted might feel. So we just took that into account.
Okay.
Okay, fine.
Good.
Very good. Ms.
Very
Thompson,
good.
what do you think? Presentation style?
Yeah, first, kudos and bonus points for going first, team. That's always a hard position to be in. I love the topic that you chose. At any time you're saying something timely and relevant, this is something that was affecting everybody, not just those people who were actually the students, but it's affecting teachers, it's affecting family members. It's important to be studying something that's relevant and pertinent. So good choice on that. I actually had a couple of questions, not so much on the presentation perspective, but as Dr. Giordano pointed out, I'm also a student. And I actually started just after the COVID lockdown began. I personally have absolutely embraced learning online. I love it. And I'm dreading going back to a school, a campus environment. And now I kind of get why. I am by nature introverted. So this is really making sense. I'd be interested to know, if you take this a step further, I'd be interested to understand within that sample group, those students lived on campus and were in a dormitory type situation versus lived locally or lived with their families. And then when you're assessing changes in their GPA, could you turn into maybe a mixed method study, go that step further and actually interview these students and find out if their GPA stayed the same or the GPAs changed, what other parts of their lives changed? Did the GPA stay the same, but their overall work-life balance improved or other aspects of their life improved? Did they put in less effort to get better grades, or did they have to put in more effort to keep the grades the same? kind of take some of those other aspects into consideration. Just some of my thoughts. But overall, this is a great presentation to kick this off with. Really, really well done.
Very good. Yeah, I was very, very pleased. Excellent job. And thank you to all of our subject matter experts for their wonderful overviews. Let's carry on. Good job. Very, very good job. Proud of you.
All right. Yes, I'm very proud of you guys as well. Congratulations. Again, that was terrific. Thank you so much for sharing all of your hard work. And then